Thank you very much for the very kind introduction and the invitation to give this talk. It's a gorgeous day to be in New York, and it's a pleasure to visit the Simons Foundation, and it's a particular honor to give this talk on emerging trends in autism. I'm going to start with the first slide that summarizes my perspective on where things are in autism spectrum disorder today. Due to many efforts from many labs, and many of them were spearheaded by the Simons Foundation, there's been a huge genetic revolution in autism field. And we believe there are approximately 400 to 1,000 different genes that are linked to autism spectrum disorder. That obviously generates a problem in terms of developing treatments. So the heterogeneity of the causes of autism is actually a handicap in some ways to developing treatments. So the way I simply think about this thing is that there might be potentially a broad spectrum treatment that is effective against all sorts of autism spectrum disorder. I think that's the optimistic scenario, and work from Mark Baer's lab, for instance, has suggested that there are certain forms of autism, such as Fragile X, that are quite different from other forms of autism, such as tuberous sclerosis. In fact, these two diseases might be diametrically opposite of each other in terms of synaptic function. So a treatment that's effective in one might be harmful to the other, and vice versa. Another scenario is that we may have to develop a different drug or a different intervention for each gene, which obviously is quite a pessimistic scenario, and I don't think we have the resources today to be able to take on this such a large endeavor. So many of us are hoping that there might be subcategories of autism that share common cellular or circuit defects. So a treatment for one of those diseases in that subcategory might be generalizable to other diseases in that subcategory. What I'd like to argue for the next 50 minutes or so is that tuberous sclerosis complex, a disease that we're working on, could be one of these pioneering diseases for one such subcategory and might be generalizable to certain types of autism spectrum disorder. The approach that we've taken at Boston Children's is very similar to approaches that have been taken in other institutions. Because we are a hospital, we start with individuals affected with autism spectrum disorder. We focus on monogenic or genetic causes of autism. We try to perform as much deep phenotyping of the individuals affected with autism with really the goal of identifying biomarkers that can accelerate our treatment trials going forward. Over the years, we've used many preclinical cellular models. In the past, these were mostly mouse and rat models of neurons in culture. But more and more, we are using iPSC-derived neurons to address this question.
What is iPSC?
Induced pluripotent stem cells. So these are stem cells that have the potential to become any cell in the body. Then we've generated a number of preclinical mouse models that replicate various aspects of the disease, tuberous sclerosis, for instance. And we've done preclinical treatment trials that have led to clinical treatment trials. One important thing to realize about this circle is that we are likely to go around the circle several times and try to come up with safer and more effective treatments as we learn from each time we go around the circle. What I'd like to do is to start with one patient I saw over 10 years ago in the TSC, tuberous sclerosis clinic, just to give you a flavor of the type of difficulties that individuals with TSC face. So this is Adam's story, and I have permission from his family to share this with you. Adam's mother had an ultrasound while she was pregnant with Adam, and this in utero ultrasound revealed growths in the heart of the fetus. This is one of the symptoms of the disease for tuberous sclerosis. So the issue of tuberous sclerosis was raised during the prenatal consultation. He was born at full term, and we did a brain MRI that showed tumors in the brain, which is another feature of tuberous sclerosis. So the combination of the heart tumors and the brain tumors gave him the diagnosis of tuberous sclerosis. I saw them when he was about 2 or 3 weeks of age with his family, and I counseled them about the possibility of the baby developing seizures in the next several months of life. They called me about 3 months of life saying that he started having seizures. In fact, a type of seizures called infantile spasms, which are quite typical of this disease. We started him on a medication called vigabatrin, which happens to be particularly effective with for this disorder. And his seizures stopped after 1 dose of vigabatrin. He now is about 13, 14 years of age. He sees several specialists at Boston Children's in our multidisciplinary program. And his seizures has been under very good control. He hasn't had any seizures after that first infantile spells that he had. However, around 3 years of age, Adam was diagnosed with autism spectrum disorder. He is minimally verbal. He has one toy that he plays with. So Adam and patients like Adam has made it clear to us that there's a high incidence of autism spectrum disorder in tuberous sclerosis population. And our research program at Boston Children's has really focused on the relationship between autism spectrum disorder and tuberous sclerosis for the past decade or so. I want to highlight some features of tuberous sclerosis that make it a particularly good model to study as a model for autism spectrum disorder. So about half of the patients with TSC will go on to develop autism spectrum disorder, which is a high ratio among genetic disorders. Where, very importantly, many of the TSC patients may be diagnosed before birth or at the time of birth because of these heart tumors that can be diagnosed. The cellular mechanisms apparent in TSC, which I'll describe in a couple of minutes, are beginning to be understood, not just from the work that's being done in neuroscience, but also the role of these genes in oncology, in transplant medicine, et cetera. And very importantly, we have FDA-approved specific inhibitors of this pathway, the mTOR pathway, which I will describe, which can be repurposed for preclinical or clinical trials. So the combination of these 4 factors make tuberous sclerosis a particularly unique opportunity within the realm of autism spectrum disorder. So briefly, tuberous sclerosis is a disease, is a genetic disease. It can be inherited in an autosomal dominant fashion. It causes benign tumors in the brain, but also in the eyes, skin, kidneys, and the heart. And it usually presents to child neurologists like myself because 90% of the patients will have seizures sometime in their life. About half of them are affected with intellectual disability, and half of them are affected with autism. Its incidence is about 1 in 6,000, so we estimate approximately 50,000 people in the United States, about 1 million people worldwide affected with this disease. And the genes have been known for about 2 decades. TSC1 and TSC2 are the causal genes. Loss of function of 1 copy of TSC1 or 1 copy of TSC2 is sufficient to give you a diagnosis of tuberous sclerosis. So the main way we diagnose patients with tuberous sclerosis these days is through MRI. Here's an MRI image of the brain of a patient with TSC. These white spots you can see are the cortical tubers. And on average, a patient with TSC can have 20 to 30 cortical tubers. If you look at this post-mortem specimen, you can see that tuber shows thickening of the cortex and lack of myelination underneath the cortex. Under the microscope, those tubers show characteristic enlarged cells. These cells can be 150 to 200 microns in diameter, about 10 times the size of a normal neuron. And around 2001, 2002, 5 different groups, research groups around the world, made a connection between TSC genes and cell size abnormalities. So this was done predominantly in Drosophila. Here's an example of Drosophila that's missing the TSC1 gene. This is the eye of the Drosophila. The whole eye is bigger. The ommatidia that make up the eye are bigger, and the hair cells are bigger if they're missing the TSC1 gene. It turns out that any cell, either in cell culture or in the animal that's missing the TSC1 or TSC2 gene, grows bigger than the wild-type cells. And the reason for that is this following pathway that's been very well conserved throughout evolution. So it turns out that the cells control cell size by regulating the amount of protein synthesis. And that is under the control of 2 genes, 2 proteins, Reb, which is a GTPase, and a kinase called mechanistic target of rapamycin. When these 2 proteins are active, they turn on a program of protein synthesis that leads to cell growth. What TSC1 and TSC2 proteins do is to form a heterodimeric complex that puts a brake on this protein Reb. So their normal function is to keep protein synthesis and cell growth under check. As you can imagine, if you are missing either TSC1 or TSC2, this whole complex is not active, and you get upregulation of Reb and mTOR activity, resulting in too much cell growth. It turns out that the cells have taken advantage of this pathway in many ways. So for instance, growth factors activate a kinase called AKT that phosphorylates and inactivates this complex, allowing further protein synthesis and cell growth. So cells grow in response to BDNF or insulin, for instance, by inhibiting the TSC complex. On the opposite side, energy level in the cell regulates this complex in a different way. So if you break down your ATP to AMP, there's a kinase called AMP kinase that gets activated by AMP levels that phosphorylates and activates this complex and allows it to put a brake on protein synthesis, so a starving cell doesn't continue to grow. One of the first questions that we asked was, what is the true incidence of autism spectrum disorder in the TSC population? At the time we started our studies, retrospective studies suggested ranges starting from 17% to 80%, depending on the cohort being studied. So Shefali Jesti, who was a fellow at Boston Children's at the time, collaborated with a group in the United Kingdom that had followed infants with tuberous sclerosis prospectively. And they had done ADAS at 2, 3, 4, 5 years of age. And when Shefali analyzed this data, what she showed was that about 20% of the patients have full-blown autism, and another 30% have a broader spectrum, autism spectrum disorder. So on total, about half of the patients have autism spectrum disorder as we would define today, and the other half are not affected. One of the questions I get asked a lot is autism due to tuberous sclerosis different than non-syndromic autism? So in my clinic, when I see a patient with tuberous sclerosis, if I don't notice the skin manifestations that patient has, I cannot differentiate them in terms of their autistic features from a patient without tuberous sclerosis who has autism. But that's just sort of an anecdotal impression. So there are actually 2 studies now that have looked at the symptoms of autism in the TSC population compared to the non-syndromic autism. So first study was Patrick Bolton's lab, where he did a principal component analysis of ADI, which is an interview-based way of looking at the symptoms of autism, in 5 genetic disorders. And he could show that by machine learning, the symptoms correlated with the genetic disorder. Then he took the principal profile, ADI profile of the patients with the genetic disorders and compared it to the AGREE sample, which is this non-syndromic autism sample. And the genetic disorder that correlated best with the AGREE sample was the TSC population. So the symptoms as measured by ADI in TSC are very similar in terms of autism symptoms to the AGREE sample. Another study done by Shefali Jesti more recently with Chuck Nelson used the ADAS profiles of infants and toddlers with autism spectrum disorder. And she compared 4 groups, typically developing children, TSC patients that are not on the spectrum, non-syndromic autism spectrum patients, and then TSC patients that have autism spectrum disorder. And what this shows are the different scores of the patients on the ADAS items. And as you can see, the purple line, the TSC population with autism, and the non-syndromic autism population follow each other very closely. Again, arguing that the autism symptoms that we see in TSC are very close to what is seen in the non-syndromic population. So why do patients with tuberous sclerosis have autism? I won't give you the answer to that at this point, but most of the literature has focused on the cortical tubers. In fact, there was a nice study from Patrick Bolton using 1.5 Tesla MRIs in the United Kingdom, where he suggested that temporal lobe tubers were necessary but not sufficient for having autism spectrum disorder. These were early studies. Since then, we've been using 3 Tesla MRI, and we were able to detect many more tubers in patients with TSC, and that correlation doesn't seem to hold anymore. And there are several other studies that have seen correlations with tubers in the cerebellum, for instance, or in the frontal lobes to correlate with autism spectrum disorder. So I think the location of the tubers or the tuber burden and its role on autism diagnosis is still out for debate. In my practice, I'll show you 2 examples of 2 patients that I see in the TSC clinic. Here on the left is a 17-year-old boy who has these 3 tubers. He has mild learning disabilities, and he's in an integrated class in high school. The girl on the right is 18 years of age. She has approximately the same number and size of tubers in the frontal lobes, and she's nonverbal and has autism. So just looking at the conventional MRI, it's very hard to predict who's going to have autism and the severity of the neurological symptoms that you see. So instead of focusing on these white spots, we've instead focused on the non-tuber pathology of TSC. And for many years, we have been using diffusion imaging to look at the connectivity on the brain. I'll come back to these studies toward the end of the talk. Of course, it's very hard to draw mechanistic conclusions from human studies. So we've turned to mouse models to investigate the role of TSC1 and TSC2 genes in neural development and function. So I'll tell you over the next 20 minutes or so several short stories about the role that TSC1, TSC2 proteins play in developing neurons. So I'll start with the role of TSC proteins in axon specification. When we first started these studies, it was clear that TSC1 and TSC2 proteins are found in the growth cones of neurites in culture, but it wasn't clear whether these growth cones were axonal growth cones or dendritic growth cones. So what we did was to take the antibodies that were available to this set of proteins in the TSC-mTOR pathway, and we stained developing neurons in culture. Here, for instance, is a hippocampal neuron. Here's the cell body, here's the short dendrites, and here's the long axon. And as you can see here, one of the proteins, the phosphorylated form of TSC2, is predominantly staining the axon, and there's very little staining in the dendrites. We saw the same staining with Reb and S6 kinase as well. So why is there predominant activity of these proteins or their localization to the axon occurring? So it turns out that one of the first decisions a neuron has to make is to determine which one of its nascent neurons will become the axon. This has been studied by several groups, especially for hippocampal neurons. So in the beginning, all the nascent neurons have equal potential to be the axon. Over the next 48 hours, one of them grows faster than the others, and that becomes the axon. If you ablate this axon mechanically, for instance, one of the other neurites will then become the axon. So the cells have an intrinsic process, intrinsic program to make sure they have an axon and only a single axon. So we took advantage of this assay. We stained neurons with an axonal marker like tau1 or a dendritic marker such as MAP2. And as you can see, we can then classify the neurons as having no axon, multiple axons, or a single axon. And vast majority of the cells in the nervous system have single axons. So what happens if you're missing the TSC gene in this assay? So we knock down TSC2 in this case. The same results are seen if we knock out TSC1. On the top is the control neurons. Here you can see a single axon coming out of the cell body with multiple dendrites. At the bottom, I hope you can appreciate this with this projection, there are multiple tau-positive processes coming out of the cell body. And there's a very robust phenotype. If you quantify this, you go from 80% of the cells having a single axon to 40% of the cells having a single axon and 40% having multiple axons. And importantly, if you treat these TSC-deficient neurons with rapamycin, the small molecule that inhibits the kinase that's downstream of TSC, the mTOR kinase, then you can reduce this multiple axon phenotype. Since this work is already published, I won't go into the details, but we took a candidate gene approach to understand why hyperactivity of mTOR was resulting in this multiple axon phenotype. We took a number of proteins that have been implicated in axon growth. In this case, one of them, SAD kinase, which has been studied by Josh Sahin and his colleagues, showed a very interesting pattern. So if you knock down TSC, the protein levels of SAD kinase go up. If you treat the knockdown cells with rapamycin, the mTOR inhibitor, you come back to the control levels in terms of the expression of this protein. And importantly, there's no change in the levels of the mRNA for this gene. So it seems like there's a subset of proteins that are regulated by TSC and mTOR at the translational level, not at the transcriptional level. And those changes in the expression of these proteins can contribute to the axonal phenotype. In fact, we showed in that same paper that if you knock down SAD kinase at the same time you knock down TSC, you can reduce the multiple axon phenotype. Now, this was a candidate gene approach. Since then, we've taken more unbiased approaches to try to understand the subset of mRNAs that are regulated by TSC and mTOR in neurons. So we took advantage of a technique that was developed here in New York. We collaborated with Miriam Heyman that has been at MIT since her postdoc. She developed this technique where you can pull ribosomes using an antibody specific to GFP, and you can identify the mRNAs that are associated with ribosomes. Using this technology, which I won't get into the details of, we have identified now a subset of mRNAs that are upregulated when you're missing TSC, a subset of mRNAs that are downregulated when you're missing TSC, and a subset of mRNAs that are regulated by rapamycin treatment in the two conditions. So it seems like there's quite a bit of heterogeneity in the protein synthesis or translational regulation that's downstream of mTOR, at least in this subset of neurons. And we're very interested in pursuing this regulation of mRNAs and protein synthesis by TSC and mTOR pathway in neurons going forward. Everything I've told you so far occurs in cells that are missing essentially both alleles of TSC1 or TSC2. But TSC is an autosomal dominant disease, so having one mutant copy is sufficient to have the disease. So we wondered whether we can see any effects on axon specification where you're missing just one copy of TSC1 or TSC2? And the answer is no. You don't get multiple exon phenotype with missing just one allele. So we decided to look at other abnormalities that could occur in the development of axons. And we turned to axon guidance. This is a field that's, I'm sure, familiar to many of you. It's been studied by various groups. Axons reach their destination by interacting with positive or repulsive cues. And some of these could be cell membrane associated, others could be secreted molecules. One of the best studied axonal pathways in the brain is the projection from the retina to its target in the brain, in this case, the geniculate nucleus in the thalamus. The advantage of this pathway is that the eyes are very accessible, so you can put different colored dyes into the eye. The projection has been mapped by several groups, so we know exactly what molecules on the axons are responsible for this topographic mapping into the brain. So we took advantage of this very well-studied pathway and studied the role of TSC1, TSC2 genes in the mapping of these projections into the brain. So here's an example from that study where we put di crystals in the retina, and we looked at the projection of these axons into the lateral geniculate nucleus in the thalamus. So as you can see on the wild-type littermates, all the axons go to this corner of the LGN, the lateral geniculate nucleus. In littermates that are missing one copy of TSC2, they go to that corner of the LGN, but they also project throughout the axis of the LGN, as you can see here. The cartoon summarizes the result. The red shows the wild-type axons, and the green shows the TSC2 heterozygous axons. So missing one copy of the gene gives you mapping defects in this projection from the eye to the brain. Why does this happen? To look at that more closely, we made single neuron cultures and stained these neurons in culture with phalloidin, which is for the actin cytoskeleton. So we can look at the growth cones of these dissociated cells in culture. So here's a growth cone with lamellipodia and filopodia. If you put a repulsive ligand, in this case called ephrin, onto those growth cones, the growth cone collapses and you get this collapsed bulb morphology. So we looked at the wild-type growth cones from the retina versus the TSC2 heterozygous growth cones. And before ephrin treatment, there was no difference in the dynamics of these growth cones. After ephrin treatment, which is this repulsive molecule that causes growth cone collapse, as expected, 70% of the growth cones collapse in the wild-type case. In the TSC2 heterozygous case, missing only a single copy of the TSC2 gene, only about 40% of the growth cones collapse. So they were insensitive to treatment by this repulsive molecule, which we believe contributes to the abnormality. I feel like I'm missing this slide. Okay. This is already published, and so I'll skip this data. But basically, we looked into the signaling pathways downstream of ephrins, and we identified that within 5 minutes of treatment with the repulsive ligand, you see downregulation of the mTOR pathway very quickly after this repulsive ligand reaches the growth cones. One of the important fields that's been, or themes that is becoming obvious in neurobiology is that there's protein synthesis that is occurring in axons and dendrites, not just in the cell body. This local protein synthesis appears to be very important in terms of function and development of the neurons. So we wondered whether the mTOR pathway that's regulated by these axon guidance molecules, ephrins in this case, could be regulated in an mTOR-dependent pathway, mTOR-dependent way. So what we did in this case was to take cell bodies, take neurons that are dissociated from the hippocampus. We put them on a small insert that has very tiny holes, such that the cell bodies cannot go through the holes, but the neurites can go through the holes. We can then scrape off the cell bodies, neuronal cell bodies, and what we're left with is just the neurites, axons and dendrites of these cells. And we can then put in S35 methionine to look at protein synthesis just in the neurite fraction. When we did this and we applied different ligands, what we found was that about 20% of the protein synthesis was being repressed by ephrins, the repulsive ligand that is responsible for the guidance of these axons. Another 30% was being repressed by rapamycin, the mTOR inhibitor, suggesting that ephrins and rapamycin can inhibit de novo protein synthesis locally in these neurites. So again, this is published work, so I'm going to go through this relatively fast with this working model. So we believe when growth factors such as BDNF are exposed to the growth cone, what happens is that you turn on ERK and PI3 kinase signaling, both of which inhibit TSC complex. that results in upregulation of mTOR, and you get more protein synthesis and growth of the growth cone. On the opposite side, when you have repulsive ligands such as ephrins interact with the ligands such as EPH receptors, through double inhibition, which I won't get into, TSC complex is actually activated. And that activation of the TSC complex puts a brake on mTOR. You get a reduction in protein synthesis, which we believe contributes to the retraction and collapse response of the growth cones. Of course, in the disease state, when you're missing a dosage of TSC1 and TSC2, the cell surface signaling does not regulate the mTOR complex, and you get exuberant growth and collapse resistance. So this is a hypothesis, and we're following this up in various ways to make sure it happens in vivo. But one of the things that we really focused on in the first two stories I've told you so far is this regulation of mTOR, the regulation of protein synthesis by mTOR. It turns out that mTOR is a very important regulatory kinase that does more things in the cells than just regulate protein synthesis. And one of the things that it regulates is mitochondrial function and something called autophagy, which many of you probably heard about recently from the Nobel Prize that was given this week. So we started looking about 2 years ago in the function of autophagy and mitochondria in TSC. Now, why did we start working on that? It turns out that a few years ago we published a paper looking by electron microscopy at the number of autophagosomes in TSC-deficient neurons. And what we had found at the time was that the double membrane vesicles that are these autophagosomes are actually increased in TSC-deficient neurons. In an in vivo study, we had also shown that there's an increase in the number of mitochondria in the brains of mice that are missing TSC. And more importantly, a different group looked at brains of patients which were resected at the time of epilepsy surgery, and they found these dense core lysosomes, all of which suggests an abnormality in the autophagy mitochondrial accumulation pathway. So we wanted to look at really what is the effect of TSC deficiency on mitochondrial homeostasis. mTOR can regulate mitochondria. In some cells, it regulates its biogenesis, but it also regulates the dysregulatory pathway called autophagy, which is the eating up of the cells and organelles in the cells. So what is the final effect on mitochondrial dynamics? This is not a published story yet, so I'm going to spend a few more minutes on this, more detail than the other stories that I'm going to talk about. So Darius Ibrahim Fakhari, postdoc in the lab, decided to take on this question. And the first thing he did was to look at the total mitochondrial mass in neurons that are missing TSC. He labeled the mitochondria with a dye. He did FACS sorting. And what he found was that there was almost a 2.5-fold increase in the mass of cells that are missing, mitochondrial mass, in cells that are missing TSC2 compared to control cells. Now, mitochondria are not just found in the cell body, but they're also found in the neurites. So he wanted to ask the question, what happens to mitochondria in the neurites when you're missing TSC? And surprisingly, what we found was that there was actually a reduction in the number of mitochondria per unit length in neurons that are missing TSC compared to wild-type neurons. We also looked at the corpus callosum of knockout mice that are missing the TSC, and we saw a similar small but reproducible decrease in the mitochondria per length in the TSC-deficient neurons. So what seems to happen is that we see an increase in the number of mitochondria in the cell body. We see a decrease in the mitochondria in the axons. So we wondered what is happening to the function of these mitochondria in TSC deficiency. So we did a seahorse assay, which is a conventional way of looking at the oxygen consumption rates in the presence and absence of the TSC gene. Just to make a long story short, we saw deficiencies, a reduction in oxidative consumption rates in TSC deficient neurons. So the mitochondria are not functioning as well as wild-type mitochondria. Now, this is in the whole cell. What happens to mitochondria in the axon? Another way of looking at the function of the mitochondria are by using these dyes that give you a sense of the mitochondrial membrane potential. And what we found was that TSC-deficient axons have mitochondria that have lowered mitochondrial membrane potential, so less well-functioning mitochondria in the axons. This was done first in rat neurons, but we want to know if the same thing happened in human neurons. So we made some iPSC, again, stem cell-derived human neurons, and we looked at the mitochondrial function in human neurons that are missing the TSC2 gene. We looked at both heterozygous neurons, and we also made a homozygous knockout of the TSC gene, And what we saw was that similar to the rat neurons, there was a reduction in the mitochondrial membrane potential in human neurons that are missing the TSC gene. So just to summarize, we see not only a change in the amount of mitochondria in the axons, but we also see a change in the function of the mitochondria. We see more dysfunctional mitochondria in the axons, both in human and rat neurons. So why do you see changes in the number of mitochondria? You can have changes in biogenesis, or you can have changes in degradation and transport of mitochondria. We don't see any changes in biogenesis, but we do see changes in degradation of these mitochondria. So this is a slide that really has been made possible by decades of work on autophagy. There are not very reliable markers of autophagy, and one of them is LC3. What Darius did was to look at the co-localization of LC3 with mitochondrial markers. So this just shows you which mitochondria are just being eaten up by the autophagic process. And what he showed was that there was actually a reduction in the number of mitochondria that were undergoing autophagy. So this process, also known as mitophagy, was reduced in TSC-deficient neurons. And interestingly, when he looked at the transportation of these mitochondria, he saw abnormalities compared to control mitochondria. So this is a chimograph that shows the movement of mitochondria. If the mitochondria are moving retrogradely in the axon, they go in this direction. Anterogradely, they go in this direction. If they're stationary, they stay in the same place. When he quantified, he saw an increase in the number of mitochondria that were being moved in the retrograde fashion. One of the things that mitochondria are important for in the axon is their role at the presynaptic sites. It seems like by buffering calcium and also by providing ATP, they seem to be quite important for presynaptic function of the neurons. And this was studied by Frank Polo here at Columbia. So we collaborated with him. He gave us some reagents to look at the number of mitochondria that are at presynaptic sites in TSC-deficient cells. And what Darius saw was that there was actually a reduction in the number of mitochondria that are associated with presynaptic sites in TSC-deficient neurons, suggesting that there might be a deficiency in presynaptic function as well. So to sort of summarize what we know so far, we see a mitophagic flux reduction. We saw reduced axonal mitophagy, and we see less presynaptic mitochondria in TSC deficiency. So we hypothesized, could we increase autophagy and mitophagy and get rid of these abnormalities? So how do you do that? You can use rapamycin, which can induce autophagy in an mTOR-dependent way. Or there are other drugs that don't go through mTOR pathway, can also increase autophagy. One of them is anti-seizure medication called carbamazepine. So Darius used both of these drugs, and what he saw was that both rapamycin and this drug carbamazepine increase autophagy, as expected. The combination doesn't seem to be able to do more than what each drug can do. When he looked at the presynaptic puncta, he showed that either rapamycin treatment or carbamazepine treatment or the combination can basically bring up the number of presynaptic puncta, mitochondria at the presynaptic puncta, back to control conditions. So we're quite excited about these new findings, suggesting that there might be abnormalities in presynaptic function in TSC-deficient neurons, and then at least two drugs, rapamycin and carbamazepine, could help reduce these abnormalities. These first three stories I went through so far all focus on the cell-autonomous functions of the TSC gene. What happens if you miss the TSC gene? What happens to that particular cell that's missing the TSC gene? But it's very likely that there are non-cell autonomous functions of TSC genes, TSC proteins in neurons as well. So we sort of stumbled onto this effect when we started studying a conditional knockout of TSC. And this is the effect of TSC on myelination. So we started working with a conditional knockout of TSC because if you delete the TSC1 or TSC2 gene in all cells of the embryo dies very early, around E10. So we started making conditional knockouts, in this case a synaptic Cre-driven loss of TSC1 only in postmitotic neurons. And what we see in this case is that the conditional mutants are born at the expected frequency. They develop seizures around 3 weeks of life, and they all die from seizures by about P60. When you look at the brains of these mice, you don't see cortical tubers, but you see a marked abnormality in myelination. So this is the control. This is the TSC knockout. Then the red staining is the myelin basic protein staining. As you can see here, in 2 or 3 weeks after birth, you get more and more myelin, both in the gray matter as well as in the underlying white matter in normal control mice. But TSC deficient littermates show a marked reduction in myelin-basic protein in the gray matter, as well as a reduction in the white matter, and we've confirmed this by EM. So the first thing we wanted to ask in this case was, could we use mTOR inhibitors as we used in cell culture and change the phenotype of these mice? So we chose two different mTOR inhibitors, rapamycin and something called RAD001, which is also known as everolimus. They are very similar to each other except for a methyl group. We first did pharmacokinetics on these drugs, make sure they were getting into the brain at the expected level, and then we treated these mice with what we thought were therapeutic levels of the drugs. The vehicle-treated mice, as I told you earlier, they died from seizures by around P60. If you start treatment at P7, so it's the first week of life, and treat them with rapamycin, 90% of the mice survive, and they did not show any seizures. If you stop the treatment at P30, about a month later, they started having seizures and started having some early mortality. We saw almost identical results with everolimus. So suggesting that maintenance of mTOR inhibition is required for the longevity of these mice. So you may ask, what happens to myelination? So here's the control brain, here's the mutant brain, and here's the mutant brain on rapamycin. Much of the myelination, or at least shown by myelin-basic protein staining here, comes back to relatively normal levels, and we've confirmed this with EM as well. Now the important thing to remember here is that the cells that deposit the myelin are oligodendrocytes, and they're wild type for TSC1 expression. The cells that are missing TSC1 in this case are neurons, and there seems to be an interaction between TSC1 deficient neurons and the wild type oligodendrocytes, resulting in this phenotype. So this seems to be a non-cell autonomous effect, and we're very interested in identifying what mediates this effect. We have some candidate molecules we're pursuing. This also raises the possibility that TSC deficiency can affect other types of glia in the brain. And we have growing collaborations with Beth Stevens, who's going to be here next month, I believe, looking at the relationship between neurons and microglia in TSC disease. Finally, I want to focus on cerebellar circuitry. I won't have time to talk about circadian rhythms. It's a very interesting area, and many TSC patients have sleep disorders. But this work was published about a year ago, and if you're interested, I'm happy to talk about it in the discussion. But one of the really important things that, as a physician that sees individuals with autism, that strikes me is that even though we think of autism as a synaptopathy, a dysfunction of synapses, there has to be some specificity to which synapses are affected. So individuals affected with autism can do many things that you require functioning synapses for. So they can eat, they can swallow, they can play with one toy effectively, but they only play with one toy. So why is that specificity for this the behavioral phenotype that we are seeing with autism? So what we've been trying to do, taking a very simplistic approach, is to deleting TSC1, TSC2 genes in subset of neurons and subset of circuits to see if those circuits give a phenotype that looks in some ways like autism in the mouse models. So one of the knockout models that's been particularly informative to us has been the knockout of TSC1 in the cerebellum. So cerebellum has been implicated in autism in many ways. There's some neuroimaging findings that suggest reduction in cerebellar volume, especially in the vermis and the white matter. There are some postmortem studies, and really the most consistent finding in the postmortem studies to date is the reduction in Purkinje cell number in those individuals affected with autism versus typically developing controls. And a very interesting study from Boston Children's about a decade ago was a study where Kathleen Poropoulos looked at newborns with isolated cerebellar hemorrhage. So occasionally you can have hemorrhage in the brain as a newborn. She only focused on the group that have hemorrhage in the cerebellum and nowhere else in the brain as detected by MRI. What she showed was that there was a 37% chance of subsequently developing autism in this group that only have isolated hemorrhage in the cerebellum. There's a lot of data in the TSC population in terms of the involvement of cerebellum. So about a third of the patients have lesions in the cerebellum. There have been two different cohorts where cerebellar lesions were correlated with the diagnosis of autism. And then there's a very interesting PET study done by Hirshigani, where he looked at individuals with and without autism with TSC. And what he showed was that there was hypermetabolism in the deep cerebellar nuclei. And just to remind you, cerebellar cortex inhibits the deep cerebellar nuclei through the Purkinje cells. So this hypermetabolism in the deep cerebral nuclei implicates a hypofunction of the cerebellar cortex. So Peter Sahin, a very talented postdoc, decided to take on this question and ask whether cerebellar dysfunction could resemble autism-like features in TSC. So this hypothesis is really not saying that everything that has to do with autism occurs in the cerebellum, but really it's very basically a way to look at the circuitry of autism-like behaviors in the mice, in which cerebellum is just one node. And what we want to do was to delete TSC1 gene just in the cerebellar cortex, in cells that are the only output of the cerebellar cortex called the Purkinje cells, and see what happens to the behavior of the mice. So to make a long story short, these mice are born at the expected frequency. They have normal weights like their litter mates. We made both heterozygous and homozygous mutants. Heterozygous Purkinje cells that are missing TSC1 gene are the same in size and number as the control cells. However, the null mutants get bigger, as you would expect from mTOR activation, and they seem to die. They drop off if you quantify them, and this happens around somewhere between 6 to 8 weeks of life. So we phenotyped these mice using the behavioral phenotyping that's available. We used this three-chamber apparatus that many of you are probably familiar with from Jackie Crawley. So in this case, you put the mice in the middle chamber, give it an option to interact with a live mouse or an object, and typically developing strains usually spend more time interacting with the live mouse than the object. In the second phase, you bring a novel animal in this third chamber, and typically developing strains usually spend more time with the novel animal than the familiar animal. So we tested our three genotypes, wild types, heterozygous, and null mutants, that are missing TSC only in the cerebellum, in the Purkinje cells, in this apparatus. So what we found was that controls spent more time with the animal than the object as expected, but our hets and null mutants showed equal amount of exploration with the animal as the object. And the time investigating is shown here. In the second phase of the study, when we introduced a novel animal, again, there was a deficit in the hets and the null mutants, as you can show here. As you can see here, the control animals spend much more time with the novel animal, whereas the hets and the mutants did not show that effect. So this suggests that missing TSC1 gene, either one or two copies of the TSC1 gene, just in the Purkinje cells, is sufficient to have an autism-like deficit in the social apparatus. We also looked at repetitive behaviors using using the water maze, water T maze in this case. In this experiment, you have a platform on one arm of the T maze. You train the animal to learn the location of the platform. That's the hippocampal task. And our animals had no deficits in that phase. And then in the second phase of the experiment, you move the platform to the opposite side of the T maze, opposite arm of the T maze. And the animal has to forget the original platform, location of the platform, and learn the new location of the platform. This is a way of looking at mental stickiness and cognitive inflexibility. And that requires cerebellar function. And our animals, especially the null mutants, show the deficit here. We also saw some abnormalities in the ultrasonic vocalizations I don't have time to get into. We did some more objective recording of these Purkinje cells that are missing the TSC1 gene with Wade Regier at Harvard Med School. What we saw was a gene dosage-dependent reduction in excitability of these Purkinje cells. So heterozygous Purkinje cells were less active. They fired less than wild-type cells, and the null cells fired even less. In terms of morphology, we saw these very abnormal axonal arbors of Purkinje cells that are missing the TSC1 gene. And in terms of the dendritic spines, we actually saw an increase in Purkinje cell spine density in the TSC1 deficient Purkinje cells, and an increase in spinal length, which has been reported in certain types of autism. So we wondered whether rapamycin treatment, which was effective in our myelination and in culture experiment, could also be effective in this mouse model. So what Peter did was to start treatment with rapamycin at P7, so before the animal shows any symptoms. And what he found was that pre-treatment with rapamycin, in this case, rescues the cell death phenotype that we see in the Purkinje cells. So mutants lose the Purkinje cells, but if you start treatment at P7, you don't see any Purkinje cell death. And in terms of the behavior, the results are shown here. So these are the vehicle-treated mutants. They don't show any preference for the animal versus the object. These red bars are the rapamycin treated mutants. They spend much more time with the animal than the object as their litter mates. So rapamycin treatment early in life seemed to rescue these behavioral deficits. So just to make a long 5 sets of stories, I guess, short here. We've shown roles for TSC in axon specification, axon guidance, myelination, mitochondrial homeostasis, and particularly in terms of Purkinje cell circuitry in the cerebellum. So our goal, of course, is not just treating mouse models of TSC, but really treating individuals affected with TSC in the clinic. I think our data from the mouse model in some ways parallels what's been proposed in the autism field, that autism spectrum disorder may be a developmental disconnection syndrome. If so, I think our data from the mouse models fit this hypothesis relatively well. But what makes TSC a unique opportunity in autism is that many patients can be diagnosed very early on in life, as I showed you earlier, because of these heart tumors. If you have a heart tumor as a fetus or a newborn, you have a 95% chance of having TSC. So that's almost a biomarker in the newborn period. And we know that these individuals that have TSC have a 50% chance of developing autism. So taking those two observations into account, we decided to ask a simple question: can we detect which infants born with TSC will go on to develop autism? And this is a question we've been trying to answer at our own institution with help from Simon Warfield Shafali when she was a fellow with us, and Chuck Nelson. We're using multimodal assessment of these infants. You're using neurocognitive assessment, MRI, spatial diffusion imaging, and neurophysiological assessment of face processing. I'll show you just very few pieces of data from these early studies. So this was a diffusion tensor imaging study of the corpus callosum in 3 groups of patients. Typical developing controls, TSC patients that are not on the spectrum, and then TSC patients that have autism. As you look at the corpus callosum here, there's really no obvious difference between these 2 groups. And if you look at fractional anisotropy, which is a measure of the microstructure of the corpus callosum, there's really no quantifiable difference between these 2 groups. However, TSC patients on the spectrum have much lowered fractional anisotropy, and as you can see, much worse microstructure in the corpus callosum. So this suggests to us that even with a single genetic disorder with a high incidence of autism, we might be able to start to dissect those patients that are at high risk for having autism versus those that are low risk for developing autism. We're very excited about this possibility, so we decided to do a multi-center study to see if this original pilot data holds. So we formed the Autism Center of Excellence Network, an ACE network called TASERN. It consists of 5 children's hospitals around the country, and we've been collecting patients born with tuberous sclerosis and doing prospective EEG, MRI, and neuropsychological testing. We've collected 150 infants so far, and we're looking at the biomarkers that may correlate with autism diagnosis at the age of 3 years. Some of the early data, I don't have the results of the MRI findings so far in terms of verifying our initial findings, but some of the early data looks very interesting to us. So one of the first things we looked at was the role of EEG in predicting seizures in TSC. So we looked at individuals before they developed clinical seizures. This was published earlier this year. There were 3 different patterns. Either you had a normal EEG and you had no seizures in a subset of children, or you had normal EEG and then all of a sudden you started having clinical seizure, or in a significant proportion of the patients, you had normal EEG first, then you started having abnormal EEG, and then you started having clinical seizures, which to us is the most helpful subgroup here. So in studying this group of infants, what we found was that the age of onset of abnormal EEG, the EEG signature that tells us that you're likely to have seizures, is around 4 months of age. Now, about 2.5 to 3 months later, you start having clinical seizures. So there's a window of opportunity about 2.5 to 3 months where you have an abnormal EEG, but you don't have clinical seizures. So we're very excited about this possibility of this particular EEG signature as a marker of impending clinical seizures, providing us a window for treatment. And I'll come back to that in a second. So in terms of treatment, what can we offer these individuals? In the mouse models, mTOR inhibitors look very promising. As I showed you, we can improve myelination and seizures in our mouse model. Michael Wang's lab at WashU used a GFAP-CRE, so just astrocyte-specific knockout of TSC, and he has evidence suggesting that these mice also develop seizures, which can be prevented or stopped by treating with rapamycin. Alcina Silva at UCLA worked with a very mild mouse model of TSC that has just learning problems, water maze learning problems, and he treated these mice with just 5 days of rapamycin and showed that you could improve long-term potentiation and learning in these mice. And then I showed you our data on the cerebellar knockout of TSC1, where we could prevent this social deficits with rapamycin. Importantly, Michael Gambello's group did essentially an identical experiment with the TSC2 gene, and they showed very, very similar results to ours. So there seems to be a relatively robust phenotype. So taking all these preclinical data, we decided to launch a phase 2 clinical trial in TSC. We did this around 2010, looking at neurocognition as the primary endpoint, using a mTOR inhibitor, in this case everolimus, which we had used in the mouse models before. We chose children between 6 and 21 years of age with an IQ cutoff greater than 60. It was a randomized, placebo-controlled double-blind trial at Boston and Cincinnati. We actually enrolled 47 patients from these two sites. We did neurocognitive testing at 0, 3, and 6 months. We also look at autism, seizures, and sleep as secondary endpoints. We just finished this trial recently, and unfortunately, we actually don't see much of an effect of everolimus treatment for 6 months on the experimental group compared to the placebo-treated group. So this is obviously very disappointing, but we've learned quite a bit from this trial, I believe, in terms of both the endpoints and also the choice of subjects to enroll in this study. So we're coming back to our discovery plan. I think we've gone through this circle once. We have to go through it, I suspect, a couple of more times. So we're trying to learn from the first time we went through the circle and come up with ways we can make these clinical trials in the future better and better. So the second trial that we're going to do, which is just funded by the NIH, is a prevention trial, because we believe starting treatment at 6 years of age might be too old for changing the neurocognitive profile of individuals affected with TSC. So we are actually going to start a medication called vigabatrin based on the EEG signature that I mentioned earlier that we found in our biomarker trial. When the children show this EEG profile, they're going to be randomized to vigabatrin versus placebo. And we're going to look at the epilepsy and developmental outcome at 2 and 3 years of age. This is going to start in November in 7 sites around the country. So I think we've learned a lot in TSC. We've recently had a workshop at NIH summarizing our recommendations going forward on research, both on the preclinical side and the clinical side. But I want to remind you that I've only talked about one disease, or 2 genes, TSC1 and TSC2, and there's approximately another 900 genes that give you susceptibility for autism. So how can we start to focus on these subgroups that I mentioned earlier that might have some convergence, subcategories? So as a part of that, we formed another consortium to look at 3 genes, or 3 groups of individuals affected with mutations. Obviously the TSC1 mutation, but when you think about it in terms of the biochemistry of the synapse and the cell, there are other proteins that are very proximal to the TSC genes in terms of regulation of this mTOR pathway. One of them is a group of individuals affected with PTEN mutations. Another one is a group of individuals affected with SHANK3 mutations, which results in a disease called Phelan-McDermid syndrome. So we formed a rare disease consortium called Developmental Synaptopathies Consortium. What we're doing is to compare individuals with tuberous sclerosis, Phelan-McDermid, and PTEN mutations in terms of neurocognitive assessments, autism assessments, as well as biomarkers. We also have a PTEN clinical trial with everolimus, the same drug that we recently used in TSC population, but this time using a biomarker, in this case eye tracking, as one way of looking at the efficacy of the treatment. So this is, I think, is going to start to give us some comparative pathobiology of 3 diseases that we hope are similar in the pathway. And we're going to start to look at differences and similarities in the 3 groups of individuals. Of note, about 5 years ago, Autism Speaks started an initiative to look at mouse models of autism. It's called PAC, Preclinical Autism Consortium for Therapeutics. It includes our group at Boston Children's, but Carrie Jones's group at Vanderbilt, and led by Jackie Crowley at UC Davis. And we've started doing this comparative analysis in the preclinical models, and we've so far analyzed 6 different mouse models of autism using repetitive cohorts, so finding abnormalities in one cohort and making sure that we can see the same abnormality in another cohort. So 2 different cohorts of mice. They are undergoing, mice and rats, I should say, two different species for the same gene. We're doing not just behavioral analysis, but EEG as well. So we are hoping some of our findings from the human studies will be informed by our findings in the mouse studies and vice versa. So I'll stop here. I'll acknowledge the people that did the work in the lab. Work on the mitochondria was done by Darius Ibrahimi. Work on myelination was done by Ebru Arjan. and work on cerebellum was performed by Peter Sai, who has his own lab at UT Southwestern. I want to acknowledge our many collaborators without whom this work wouldn't be possible, and our funding sources. Thank you very much.